Hello, I'm Masanori Matsuzaki, professor at Cellular and Molecular Physiology. Mr. Otani predicts what his opponent will do and decides what he does. Then he executes accurate desirable movement. This process is updated by learning. In the same way, we think, make decisions, and act every day. My lab is working to clarify this process in the brain. We now focus on the neural circuit for decision-making and motor learning. External and internal information is integrated in the prefrontal cortex. The values of some action candidate are compared in the basal ganglia and the prefrontal cortex. Then the action that is predicted to be the best is selected. The information then travels to the basal ganglia and the cerebellum. As a result, signals are generated to execute the selected movement accurately and quickly. The final motor command is sent to the spinal cord to coordinate the muscle contractions. The outcome of whether the movement went well or not updates the neural circuit through synaptic plasticity. We are now examining the corticocerebellar circuit and the corticobasal ganglia circuit in the mouse. We were the first in the world to develop a motor learning task for head-fixed mice using their forelimbs. We trained the mice to pull a lever to get a water reward. During learning of this motor task, we conduct total calcium imaging of all layers of the secondary and primary motor cortices. We also image long-range thermocortical axons that transfer the information calculated in the basal ganglia and the cerebellum to the motor cortex. We modify this lever pull task to examine the neural mechanisms regarding the decision-making for movement, whether to act, what to act, and when to act. From the imaging data, we extract the activity of many neurons and relate them to behaviors by using statistical and computational models. We also use optogenetics to activate or inactivate specific subsets of neurons to change the behaviors. In this way, we find the functions of each cell, each layer, and each projection in motor learning. So far, we have clarified layer-specific dynamics of neural activity during motor learning. We found that layer 2-3 neurons in the primary motor cortex are important for flexible adjustment of movement. Layer 5A neurons well memorize the motor kinematics. The basal ganglia send the kinematic information to layer 1 in the primary motor cortex through the motor thalamus, while the cerebellum sends the coordination signal required for the successful movement to the layer 3 in the primary motor cortex. We are working hard to rebuild the brain circuitry of motor control and motor learning at the single-cell level and population level. Second, we developed the imaging and optogenetic methods for behaving common marmoset. The marmoset is a small New World monkey and has many types of social behaviors such as vocalization. As you can see, the marmoset can learn to use the manipulum to move the cursor to the target above or below the fixation area on the monitor. We are now imaging the broad cortical area, including the parietal cortex, primary motor cortex, and premotor cortex. We have established total imaging of behaving animals with the widest field of view in the world. My laboratory is the only lab in the world that can perform total calcium imaging of behaving marmosets and optically control their movement. I'd like to clarify what brain functions evolved from rodent to primate and what are the circuit principles of higher brain functions in primate. We are now examining the inter-ever interactions that underlie working memory, decision-making, motor learning and execution, vocalization, and imitation. These are all novel and exciting projects. I believe that studying these interactions in marmoset will surely help us to better understand human intelligence and neurological disorders. If you want to be a pioneer in neuroscience, please join my lab. If you are interested in our marmoset project, you may be able to join my lab in REEC and CBS Center for Brain Science. Thank you.